So thank you. It's a great pleasure to be here, and I would like to, okay, I'll start again. So I'd like to start by thanking John for the kind invitation for presenting at this meeting. And I will just start by giving a brief introduction about different levels of 3D genome organization. And so as you all know, gene regulation occurs from the level of specific sequences all the way to modifications of the DNA or the histone, the nucleosomes, their modifications, but also at higher levels of gene regulation, including the contacts between regulatory regions called enhances and promoters, which form cell type specific structures, which themselves become organized in topologically associated domains or TADs, which themselves then contact with preference for contacts between active regions with other active regions or inactive regions to other inactive regions. And these then establish what is called the compartment A and B of chromatin. But genome architecture is regulated all the way to the levels of all chromosomes, how they are positioned in the nucleus relative to specific nucleobodies such as the nucleolus, and also the preferential positions of chromosomes relative to each other. And in my lab, we are interested in, first of all, developing technologies that allow us to describe 3D genome topology quantitatively across genomic scales. Also, how to understand then how are chromatin contacts regulated and how do they affect expression. And in particular, in the context of today's meeting, how do mutations associated with disease, very often occurring in chromatin regulators or in the mutations in the regulatory regions themselves, how do they affect gene expression? And so we know, for example, if we compare the promoters and enhances that are active in different parts of the brain, we see that there are, as we all know, many genes that are differentially expressed. But if we do the same exercise for the regulatory regions, the enhancers, then this number is much more diverse, indicating that the local regulatory landscapes of gene expression are highly cell type specific. We also know that in response to neuronal activity, there's dynamic changes in the interactions between different parts of the genome as we go from basal to activation and to reactivation. And in the context of disease, for example, if we take genes associated with intellectual disability, we have almost as many genes associated that are chromatin regulators involved in controlling chromatin binding, the modification or the remodeling, as many as, for example, we have genes involved in synapse organization and regulation of synaptic activity. And a final point here, that many of the mutations that are found in association with complex neurodevelopmental diseases are often found not in particularly at the coding regions of proteins, but more so within the non-coding space of the genome, and therefore thought to influence enhancer function or other aspects of higher order chromatin regulation. And so in my lab, we, and in this topic in particular, we are interested in understanding the dynamic changes in 3D genome folding in different brain cells. And the first question, and this will be the talk, most of the talk of today, we're interested in understanding how specialized are 3D chromosome structures in terminally differentiated brain cells. And here we have a number of challenges. First of all, the brain tissues are highly complex with many different cells mixed up. We don't particularly want to extract them and dissociate from their context to avoid disturbing their function. And through the talk today, I will show two discoveries that we have made with the work we've been doing, comparing different brain cell types. One of them is the discovery that long genes extensively discondense in brain cells, in particular in neurons, where this decondensation is proportional or highly correlated with the gene activation or chromatin accessibility. And we also discover specific hubs of genes that come together in a differential manner in specific neurons, for example, PGNs and DNs. At the very end, I'll just show you a snapshot of the work we're now developing, which aims to understand what are the alterations in 3D topology associated with neuronal disease, and if we can map these topologies in detail and understand how they relate with gene regulation, can they help decipher the mechanisms of disease? And here we're working at the moment with several knockouts or environmental interferences. 3 of them are SADP2, ATRX, and CTCF conditional knockouts, specifically in the PGNs, all of which, in all of these mouse models, we have alterations in memory formation. And to do this, we have developed a method called genome architecture mapping, which combines aspects of imaging with genomics. And in this method, we start with the ultra-thin cryosections. They're called Tokoyasu cryosections, where tissues or cell pellets are fixed very well, as for electron microscopy, to preserve all the structure and also the content, not only of the genome, but also the RNAs and proteins that characterize the cell type. After fixation, the cells are simply embedded in a cryoprotectant, which is just a sucrose solution in PBS. They're frozen in liquid nitrogen and then cut into very thin sections using an ultracryomicrotome, which is common in most of the electron microscopy facilities around the major institutes in the world. And initially, as a PhD student and then in my own lab, I use these sections, namely to probe the structure of the genome, especially to understand the organization, in this case here, of chromosomes, whether they're mixed or not, also to be able to localize the position of specific molecules, such RNAs or proteins, and all of this while retaining exquisite structure that can be achieved in electron microscopy. However, these imaging methods, especially 10, 20 years ago, they were essentially quite difficult because we're always only looking at one or another chromosome. We can map very few molecules at the same time, and it was very difficult to have a global understanding of what all parts of the genome were doing, to understand, for example, if there's a particular contact that's observed, is it really the driver important specific event, or it's actually a bystander of some other aspect occurring in the nucleus that we are not mapping in that moment. And so to avoid having to rely on microscopy or in the use of probing only a few parts of the genome at a time, we developed a technology called genome architecture mapping, which essentially starts with the same exact preparation, but instead of moving these sections to a glass cover slip, for example, for confocal microscopy, instead we move them to the laser microdissection microscope, where individual nuclear slices or cell slices are laser microdissected into individual tubes. And with the advent of single cell whole genome amplification and next generation sequencing, it became then possible to extract the DNA content within these nuclear slices, to then use bioinformatic methods to understand which parts of the genome are present or absent, and from this data to build a number of various different aspects of 3D genome topology. So how does this work in a schematic way? So here we have a very schematic picture of the nucleus and 3 different parts of the genome, let's say. And it's relatively easy to appreciate if the blue and the pink loci are very close in physical space in the nucleus, that if we challenge this object with thin slices, that very often the more physically proximate loci are going to be found very often in the same slices. Conversely, if 2 loci are very distant in physical space, then it's very rare, or at least more rare, that they're going to be found in the same slice. And let's say even less for the 3 of them to be found in the same slice, which is really the plane of this image. So what is the GAM data? The GAM data is really just, if I may say, a collection, a table, a very large table, where in the columns we have all the slices that or cells that we have extracted DNA from. And in the rows, we have all genomic regions, in this case, only A, B, and C. And out of this map of zeros and ones, we can then extract a number of different features. We can extract the radial position, the chromatin compaction, and in particular, what most of us are more used to, which is the pairwise chromatin contacts. And to help you understand this technology, one thing that we like to do is to just show a movie of us accumulating the data. And you can see that if we collect 13, this looks very noisy. There's already some patterns emerge, but because there's only 13 slices, we cannot be sure of them as in any other area of science. There's also a lot of windows of pairs of genomic windows that have never been found together in the same slice. And we don't know if this is because of statistical undersampling or because they're actually so far away that they never come together. And so as we increase the size of our sample, this is now 33 slices, you can see this is getting better. Oops. And I clicked in the wrong place. And so now let's say 53, 63, and you see that now the patterns are refining. Most windows are now actually occupied with the events where the two genomic regions, even though very far apart, they statistically found sampled in the same slice. And as we continue, you can see that then the patterns refine, and some patterns just become, they stay there because we had already sampled enough, but others are tuning exactly the value. And you will see that after about 200, 300, it was fairly stable, and that we like to collect between 500 and 1,000 to allow us to achieve, robust statistical conclusions for the aspects of these patterns that we study. And also to allow us to use the lowest possible genomic resolution that each data set enables. Now, this method has a number of advantages. One is indeed the low cell number. We typically use around 1,000, between 500 and 1,000, but topology can actually be studied from even larger, even smaller data sets. Another advantage of this method is because it's very rooted in electron microscopy. It starts with very small sample sizes, and so it's suitable for small tissues, for example, in the brain, or also rare cell types within a biopsy, for example. I mentioned we can extract radial position and chromatin compaction, but also we're now moving to adding further layers of information, for example, by quantifying the transcriptome or the presence, abundance of proteins or post-translation modifications at the single cell level within these data sets. And with this, we aim to extract not only cell type, but in particular cell state information. And so for today's talk, I really wanted to showcase a work that we have been performing for a number of years, which has just been accepted for publication, and where we essentially wanted to ask a very simple question: how specialize our 3D genome structures in thermally differentiated cells? We took three brain tissues, the cortex, the CA1, and the VTA. From each of them, we took one cell type. And the challenge, as you know, in these complex tissues is that we have a number of different cells mixed up. To overcome this limitation, we added one extra layer to our GAM pipeline, and this was to immediately after the cryosectioning, as the samples are overlaid in the laser microdissection support, to add one step where we use antibodies to select, essentially to immunodetect the cells of interest. And at the laser microdissection, we only then isolated the cell type that we were interested in. And this was the only change in our original pipeline, published in 2017 by Robert Bigre and our collaborators. And after this, the pipeline is essentially the same. Now, for this proof of principle, where we wanted to ask how different are the topologies in different brain cell types, we took oligodendroglia from the cortex, pyramidal glutamatergic neurons, or PGNs, from the CA1 hippocampus, and dopaminergic neurons from the VTA midbrain. And for comparison, we took embryonic stem cell GAM data, which we had previously collected. Just to begin looking at these matrices, we collect very beautiful matrices. This is a region containing the protocadherin locus in chromosome 18 of the mouse genome. And we saw in ES cells the typical pyramidal organization of TADs. Interestingly, as we looked at the same region in the different 3 brain cell types, we could see that as the protocadherin alpha and beta become expressed in the brain cell types, we see the formation of long-range contacts between the alpha, the beta, and the gamma, which appear in all cell types. But interestingly, in the PGNs, we see the appearance of these very specific long-range contacts that connect the 3 alpha, beta, and gamma clusters with FGF1, which is expressed more highly in the oligodendroglia. Now, because we can collect these data from single mice, we did produce independent data sets, either from litamates or even from mice with different genotypes at different times. And we could indeed reproduce these long-range contacts within the protocadherin cluster genes. Moreover, we also looked at the data at a much longer range scale, here at the scale of almost all chromosomes, where we identified also the appearance of very strong long-range contacts, which actually very often connect sensory receptor genes, which are repressed in all these cell types, including the ES cells, but somehow their repression in ES cells does not coincide or require the formation of these long-range contacts. Now, after these initial visual inspections, just to understand whether there were differences worthwhile studying, we then moved to more quantitative analysis, where we essentially started looking also at specific genes, in particular at the organization of topologically associated domains. And here is one interesting locus containing the cluster of sodium channels, in particular SCN2 alpha, which is most highly expressed actually in the PGNs, and is associated with autism, epilepsy, and seizures, and actually different mutations in this gene leads to different diseases and also different onset of the disease. You can see, for example, here in the oligodendroglia, where the gene is also active, that we go from having a very strong presence of contacts, so the locus was compacted, to now having insulation between 2 adjacent domains, which where the SCN2 alpha gene is exactly at the border of this, in this position. And so with this kind of image, we could start to see that this rewiring of chromatin structure is actually that there is extensive rewiring of chromatin, but it can very often also be associated and coincide with the presence of highly expressed genes specific of the cell type of interest. And to then move to a more unbiased approach, we essentially computed the genomic coordinates of these TAD borders, and we compared them across the 4 data sets. And we could see, surprisingly, that actually in this comparison of 4, only 4 cell types, that only 14% of all detected TAD borders are actually conserved. More often, the TAD borders are specific of cell type or of brain cells or of neurons, and there are other combinations that are less abundant that we don't represent here. So because we had seen that often these borders can coincide with interesting and important genes, we then decided to do a gene ontology for all genes that were found at the borders. And this is just an example for oligodendroglia and PGNs, but it was true for the other 2 cell types, where we could see that at these new borders that are cell type specific, here in the oligodendroglia or specific of PGNs, we could see in the oligodendroglia appearance of genes at borders at genes important for synapses organization, for example, or in the PGNs, that borders appeared de novo at genes important for cognition, and these borders were only seen in the PGNs and not in the other three cell types. Now, another observation as we looked through the data was that very long genes, many of which are associated with neurodevelopmental or neurodegeneration disease, that when they were most highly expressed, that high expression coincided again with massive reorganization of the locus, in particular, often by a loss of chromatin contacts, which you can see here in the matrix in the two cases. And we wanted to understand whether there was a particular preference to this decondensation more at the promoter or the end of the gene, as polymerase transcribes through the coding region of genes. And to do this, we plotted these cumulative plots where we essentially compute the insulation as we move a diamond across the gene from the TSS to the TS. And you can see indeed that in the neurons, and also a little bit in ES cells, that the most expressed genes, they tend to have be more insulated to decondense, but this is most striking in particular as the polymerase or as we walk through the coding region of the gene at the very end, only in the two neuronal cell types, but not in oligodendroglia or mouse embryonic stem cells. Now, we wanted to study this decondensation of long genes in more detail, and to do this, we developed a pipeline called Meltron, which essentially computes the decondensation. I can explain this during the questions, and applies a statistical test. And so we have developed this domain melting score or gene melting score, which is essentially a p-value of the difference between between the two maps. And when we applied the Meltron pipeline to a comparison between each of the brain cells with mouse ES cells, we could see indeed very often that many genes are melted to a very low p-value, and that the genes are different between different cell types, although the same gene can melt in more than one cell type. And interestingly, that in particular in the two neuronal cell types, that the melting coincides with the genes being more highly transcribed than other long neuronal genes that are not melting in that particular cell type. Now, importantly, because we had these replicates between two different animals, we also compared whether the same genes melt and whether this p-value actually correlates between the two replicates. And indeed, we found that there is a correlation even in the extent of melting found in the two replicates, with many genes melting to the same extent. There's also interesting genes that melt only in one of the animals, and this we don't know if it's also to do with individual variability and something we wish to study in the future. And so we find that only show your expression, but the same patterns were also true for high chromatin accessibility. And I should say that some genes only have high chromatin accessibility and melt, and that the expression per se, at least mRNA expression, which is being quantified here, is not necessary for the melting to be observed. Now, we wanted to move from this genome-wide and bias approach to looking in more detail in events of melting. And we started with RBFOX gene here, which not expressed in ES cells, most highly expressed in the PGNs, where we see extensive decondensation throughout the coding region, but in particularly at the promoter and upstream regions. And we wanted to look at this with independent methods. And so we chose fluorescence in situ hybridization to understand whether this melting could be visible using microscopy. And as expected in ES cells, we found this dot of typical seen in fish for single loci, whereas in the PGNs, where the gene is melting, we could see, just really looking down the microscope, these extensive events of chromatin decondensation. And I note here that these objects extend more than 1 micrometer. And we also measured the area, and we could see that a large proportion of the loci are indeed melting, not all. It's also then going to be important in the future to understand whether this is one allele or both alleles, etc. As you possibly noticed in these images, in the ES cells, the ERB-FOX1 typically is at the periphery of the nucleolus, which you can see here in the quantification, or at the periphery of the nucleus, at the lamina, whereas in the active state, in the PGNs, it's most abundantly present away from these two typically repressive domains. Now, we also took another approach, and this was polymer modeling, using now a gene that melts between mouse embryonic stem cells and dopaminergic neurons. And you can see here the colors of the legend of the genomic regions. And so you can hopefully see that, for example, this extensively decondensed red, and in particular the green domains, that they are quite condensed in the ES cells, but extensively decondensing, looping out in the DNs where the expression of the gene is most abundant. And these are just two examples of the polymers that were modeled from the matrices themselves, but you can see here in the quantification of all the ensemble of polymers that indeed most polymers, and this is only for the green domain, but we saw these events also for the regions, for the red in particular, that most of these green domains are extensively decondensing. And so the questions that one wishes to then follow from this is really, what are the factors that retain the structure? What are the factors that promote the decondensation? Which genomic regions do these factors act on, exactly within this complex polymer? Where is the action occurring? And if we look across the genome, can we understand why some genes are rewiring and others not? And this would, of course, also be helpful to understand different mechanisms of disease that affect different genes in different cell types in different states. So to explore more mechanistically how the different contacts are established in these different cell types, we next focus on the two neurons, the ENs and PGNs, and we started comparing side by side these matrices. So you can appreciate here a region where in PGNs we have a lot of contacts, both more locally but also more far away. This is a 5 mega region, and in the DNs this is much more decondensed in the long range. And to do these comparisons, we started focusing really only on the top most differential 5% contacts, after essentially subtracting the matrices one from the other. And the question was, are these 5% most differential contacts essentially noise, or do they actually contain some evidence of regulation and of specificity that is typical of the cell types where we find these most cell type specific contacts? To do this, we then integrated the differential contacts in the PGNs and the DNs in blue and green with attack-sec regions that were extracted for the same cell types from available or from data that we produced in the lab, to then ask, can we find within these contacts attack-sec peaks for transcription factors that are potentially driving these cell type specific contacts? Now, there are many transcription factors that are expressed in the two cell types. As a first approach, we decided to focus only on the TFs that are more highly expressed in one or the other cell type and are represented here. And it was only for these transcription factors that we then searched for whether they were present, their transcription factor binding sites were present within the attack-sec regions. Now, this is one example. Again, we have these very long-range contacts here. You can see the attack-sec that in the DNs we have, in the PGNs, we have many more attack-sec peaks. Not only this, but we also find that they very often coincide with TFs, binding sites of the TFs that are also expressed and upregulated in PGNs. And this is not the case for the TFs that are upregulated in the DNs. We went on next to ask, do these differential contacts hold any genes of interest or just structural contacts? And remarkably, we felt about half of all the upregulated genes in PGNs relative to the DNs, they are found within these differential contacts, the most stronger differential contacts, and they typically associated with TFs, NeuroD1, NeuroD2, as well as some other of the differentially expressed TFs. Some have CTCF on both sides, but not all. And when we then did gene ontology for these, the genes that fall on the differential contacts, we could find in the PGNs, for example, genes involved in synaptic plasticity or glutamate receptor binding. For the DNs, we found addiction and other types of genes. And this is just a diagram to show you that the two upregulated genes, DLG4 and SHIZA6, form these very strong PGN contacts that don't exist in the DNs, but they're not alone. They're often surrounded or in regions where other active genes are also active, but not differentially expressed. I don't show the end result, but just to summarize, in this diagram, we found hubs of genes that are in very PGN-specific contacts that are not occurring in the DNs, and this typically involves synaptic plasticity genes, as well as some other genes listed here. Whereas in the DNs, the enrichment was found for addiction pathway genes, and this is very interesting because, in particular, the DNs in the VTA are the first responders, for example, after a single treatment with cocaine, for example. And so the contacts specific of PGNs, they are enriched for binding sites for differentially expressed genes. These hubs have genes with specialized neuronal functions, and just an anecdote that, for example, EGR1 upregulation in the PGNs coincides with increased contacts of the EGR1 locus itself, which contains EGR1 motifs, suggesting also events of self-activation. And so in summary, we see extensive specialization of genome architecture in terminally differentiated neurons and other cells. We, in the study, discovered the melting of long neuronal genes when they're most expressed, hubs of neuronal specific genes, and as I mentioned in my introduction, stricter B compartmentalization of sensory receptor genes specific in the brain cells and not in ES cells. And this study is been accepted, but it's also available as a bioarchive that you can find publicly available. Now, just as a snapshot of where we're moving on, in particular in disease, we currently or have produced GAM data for conditional knockouts of important chromatin regulators associated with disability, with learning disability. These knockouts, they commonly have the learning disability, but they also have different phenotypes, as you know. And in looking for the chromatin contacts, we're really interested in understanding whether there are some common pathways that are occurring and what are they. And these are really just snapshots. We really have only recently finished collecting this data and now deeply analyzing it. But just to show you two highlights, here is a region containing the CFOS gene, and we can see that in both the SADP1 knockout and the ATRX, there's extensive reorganization of the contact, in particular with the locus becoming covered with more dense contacts. Interestingly, also in the BDNF locus, we can also see aspects that are somehow common between the two cell types, a strengthening here of the contacts and there, but also aspects that are only seen in one of the mutants. And I don't show the CTCF, I forgot to say, because we are still collecting the second replicate. And so just to finish, to thank my lab, the brain work that I showed in the published bioarchive was led by Warren Winnick with Isa, Sasha, and also with essential and important contributions from Luna, analyzing and producing single cell attack and analyzing single cell RNA-seq data, and Dominic with the melting of genes and all the integration and analysis that work involved. For the last part on the ATRX and SATB2 knockouts, I think Isa, this has been one of our major PhD projects, and Sasha for all that work. And in blue, all the colleagues from the lab and alumni that have somehow contributed to the work, and also our collaborators and both in the cell type specialization, but also George, Nathalie, and Bonkyun for being so generous, sharing their knockout models. And with this, I finish, and I thank you for your attention.